Hello, good afternoon. My name is Dr. Nick Moore, and I'm the Associate Director of the Clinical Microbiology Laboratory at Rush University Medical Center. And I'm thrilled to be able to welcome you virtually to our microlab. And I'm also equally thrilled that you're considering Rush and the John H. Stroger Hospital of Cook County for your joint training in ID fellowship. So let's take a step inside the microlab, and I'm going to show you a little bit about what we do here in the microlab, and how you'll be interacting with us over the 2 years of your fellowship training if you come to Rush. Come this way. So this is the heart of really where everything starts in the microlab. This is our specimen processing and receiving area. So this is where all of our cultures actually get set up and plated to different media to support growth of microorganisms, whether it's a bacteria, a fungus like a yeast or a mold, all start kind of right here. And from here, they then get put into incubators where they spend several days or perhaps weeks, depending on the organism we're trying to cultivate, before final results will be entered. Once we do see growth on plates, though, the workup is divided equally amongst our benches based on specimen source. So today you're going to join me on our some walking virtual micro lab plate rounds, and we're going to see what's growing in patient samples today. So come this way. So our first stop is the blood culture bench. This is where patients have blood collected into bottles that are loaded onto a continuous analyzer, and we're checking for growth of primarily bacteria or yeast. Once a signal is detected on the instrument, a medical laboratory scientist will pull the bottle, do a gram stain where we're able to visualize the organisms and see them under the microscope, and from there we take an aliquot of that blood culture broth media and put it onto plates. And those plates then get incubated, and the next day we're able to observe microbial growth. So this is an isolate of a patient that is growing on a chocolate agar plate. We see nice yellow looking colonies, and here is the same patient's organism growing on another plate called a blood agar plate. This organism, again, is strongly beta hemolytic, as evidenced by the clearing around the colonies. And on the gram stain that we don't get to look at virtually, but you'll see on the screen here, is showing gram-positive cocci in clusters. This lets us presumptively identify this organism as a potential staph species. But the work doesn't stop there. We're able to do some very quick and rapid testing to confirm that identification. So the first step in identifying a potential gram-positive cocci is to do something called the catalase test. Here we take a drop of hydrogen peroxide and put it onto the slide, and then we're going to grab a little bit of the colony growing from the plate. We're going to scoop it up with a little stick, and then we're going to rub this into the hydrogen peroxide. And what we're looking for is this nice bubbling reaction. This indicates that the organism is producing the catalase enzyme and is catalase positive. This definitively confirms that this isolate, this gram-positive cocci, is in the Staphylococcus species. Now, if we want to know what species of staph this is, we have to do another test called a coagulase test. This test involves utilizing a latex-coated reagent with a specific antibody specific to an enzyme called coagulase. Again, we take a drop of this reagent, put it on this on this paper, and grab some of the organism growing from the colony again, and we're going to now emulsify it and spread this out into our reagent. And what we're going to be looking for is a clumping or an agglutination reaction. And this occurs very quickly, and you can see it happening already here. The suspension these particles have fallen out of suspension, indicating that there is a strong antigen-antibody complex being formed. And this indicates a positive reaction. When we see a gram-positive cocci that is both catalase positive and coagulase positive, this lets us know that the isolate is Staphylococcus aureus. This is a quick and rapid way for us to identify Staphylococcus aureus, and in fact now we've definitively identified this patient's pathogen in their bloodstream. Now, you all as prospective ID fellowship applicants know the importance of Staph aureus as a pathogen in patients, and in particular when it's isolated in the blood. This is one of the rapid ways we can go about identifying it, but our work is not complete. What we still have to do is perform antimicrobial susceptibility testing. Unfortunately, I can't wave a magic wand and show you that step very rapidly as it's done on an automated instrument. And susceptibility testing does take 18 to 24 hours to perform. But we will see those results the next morning, and that will tell us if this isolate is methicillin-resistant Staph aureus or a methicillin-susceptible Staph aureus. And that information is critical for you as an ID physician to be able to care for your patients properly. Now that we've seen the blood culture bench, we're going to go on to our next bench. Hey, Patty. Anything for micro rounds today?
I have a possible pathogen.
This is our respiratory bench, and Patty is one of our senior medical laboratory scientists. She's working up a sputum specimen that is growing multiple organisms. Here we have the chocolate agar plate again that you saw previously. Here we can identify multiple different morphologies of organisms growing. There are these green colonies, these pinpoint white colonies, and then the heavy or predominant organism, which is this flattened gray colony here. If we look at the same patient specimen on the blood agar plate now, we're going to only see really two colony types. We again see the green organism and we see these white pinpoint organisms. So what happened to that third gray predominant organism? Well, that's a fastidious organism that's only growing on the chocolate agar plate. When we think about respiratory specimens and fastidious microorganisms that grow on chocolate agar only, we're really concerned about some of the more fastidious gram-negative coccobacilli. This was ultimately identified as a Haemophilus influenza species. And that is an organism that can cause lower respiratory tract disease, including pneumonia, most often community-acquired pneumonia. And we do see a fair amount of that here in patients at Rush. Again, this is an organism that's clinically significant, so antimicrobial susceptibility testing does have to get performed when we identify Haemophilus influenza in clinical specimens. Unfortunately, we don't get to use our automated methods for doing antimicrobial susceptibility testing with Haemophilus influenza. We instead do a much more simpler and manual method known as the disc diffusion test. Here in this test, we take filtered discs that have a fixed concentration of an antibiotic and place those onto an agar plate where an inoculum of the organism has been spread out. And after overnight incubation, we're able to read those zone diameters that correspond to the amount of inhibition of bacterial growth as the drug diffuses out into the surrounding media. I have an example of disc diffusion testing that was done today. However, this is not the patient sample we just looked at. This is a patient isolate of Pseudomonas aeruginosa. It's one of the more fun microorganisms that we see in the microlab due to their ability to produce very unique pigments, such as the nice dark green pigments you can see here on the plate. This was collected from a patient with cystic fibrosis. These patients often have increased secretions and mucus production, which are really inhibitory to our automated methods for susceptibility testing. So again, we use the simpler method of disc diffusion. On this plate, you can see where there's growth of the organism represented by all of the green, and you can appreciate nicely the different antimicrobial discs that are present on the plate that have been added. And following overnight incubation, hopefully you're able to see, if we hold up the plate to the light, where there is inhibition of growth of the Pseudomonas as a result of the drug diffusing throughout the media. So our technologists will take these plates, measure that zone diameter, and look to see whether or not that zone diameter corresponds to an interpretation with that antibiotic of being susceptible, intermediate, or resistant. As a general rule, the larger the zone diameter, the more likely something is to be susceptible. This isolate of Pseudomonas was susceptible to the drug piperacillin tazobactam, which is shown here in this antibiotic disc. However, this isolate of Pseudomonas aeruginosa was also tested against the antibiotic levofloxacin and was found to be non-susceptible. It was resistant, as represented by the fact that the zone of inhibition of growth is much closer to the edges of the paper disc. So I will say in the clinical microbiology laboratory at Rush, we are a full service micro lab, meaning that we do everything from bacterial cultures and bacterial susceptibility testing, including anaerobic bacteriology and culture. We also perform fungal cultures, both yeast and molds, and perform antimicrobial susceptibility testing against yeast isolates. In addition, we also perform testing for mycobacterial species, such as Mycobacterium tuberculosis. We also have a very comprehensive molecular microbiology section in our laboratory that supports primarily our transplant infectious disease patient population, as well as patients living with HIV and hepatitis viral infections. Much of this testing is performed in a batch manner due to the nature of the testing and the instrumentation that we utilize, but we do have some rapid options for testing available for other viral targets or pathogens. Over the course of your 2 years of fellowship training when you're here at Rush, you'll be joining me like this, albeit in person, twice a week for plate rounds when you're on ID inpatient consultation services with either our general or our transplant infectious disease teams. Additionally, at the start of every fellowship year, we conduct an intensive 2 to 3 day microbiology boot camp where you get some crash course lectures as well as time to come into the laboratory and to get your hands at doing some testing, including streaking plates, doing some gram stains, and taking a shot at interpreting those gram stains and seeing how you do. Also, over the course of your fellowship training, you will spend 2 weeks here as in an elective rotation during your second year of fellowship training, where you will actually get to come up to the laboratory where you're relieved of clinical inpatient responsibilities and spend time rotating through each of the benches with several of our senior medical laboratory scientists and getting to take a deep dive into our culture and bench procedures so you really understand the work that goes on in the clinical microbiology laboratory. You'll also have opportunities to interact with us over the course of your training when you rotate on your antimicrobial stewardship rotation with Drs. Juan and Shankran and our talented clinical pharmacy specialists. And there are opportunities to engage in research related to clinical microbiology and diagnostic stewardship or new test evaluations if that is an area of interest of you. So with that, I think we've concluded our virtual micro rounds. Again, thank you so much for your time. I'm sorry we weren't able to do this in person, but I look forward to seeing each of you in July if you select Rush for your fellowship training. Thank you.